We're doing this by starting with data, so real patient data. Here we've collected a diverse database with our clinical partners regarding pathology, ethnicity, age groups, and also we're including animals in this. With that, we can then create three-dimensional representatives of the population, so this so-called virtual population. This can then be combined with virtual benchtop testing and also advanced anatomical simulations. So as a first step, we can provide here statistical population analysis to better understand the patient cohort. So create rationale for worst case assessment and define inclusion-exclusion criteria, because you're not developing your device just for the average patient, but also then you want to define these critical areas. And as said, this can then be combined with this anatomical simulations that will show then later on how this works in detail. And finally, you create digital evidence can be used then for the regulatory submission, where we are compliant to the guidance document that I've just shown you. So how does this look in action? So here you can then, as a first step, select the patient cohort. So create your virtual patient based on your target group that you want to address. You can then upload your own device, include this then into the patient. So for example, here first position it at a certain location, and then in an easy environment, you can select then also the conditions that you want to have for the simulation. The next step, you can then access also the statistical analysis. So as such, I define maybe the worst case assessment. Like here, then a multitude of patients, you can see, you can cluster it in male, female patient, but also in different pathologies, diseases, which are measurements that you perform there or that automatically are performed, to then have this density curves also to access like the statistical of the population. Then this can be combined then with non-clinical assessment, like here, for example, to assess the hemodynamics of a heart valve, and also fatigue analysis of a stent, combine then also the two different isonorms, or also the cannula pressure drop analysis to assess hemolysis or blood clotting. And then as a next step, this can then be performed and combined with the anatomical data, like here with an LAL occluder device, but also the catheter application. This is an example I want to show next in more detail. And then finally, also what is possible is to use electrophysiology to even include the beating heart in the assessment there under different conditions.